Thank you, Brenda. I'm pleased to once again be a part of this important workshop organized by the Office of Generic Drugs and the Office of Pharmaceutical Quality in collaboration with the Office of Small Business and Industry Assistance. There are few things as important to our work in healthcare as stimulating and supporting innovation in generic drug development. To this end, I also want to thank the many developers and other stakeholders who've joined today's discussion, as well as the support provided by industry to the generic drug development program to help make the program possible. The word innovation is an important one, and while there's sometimes a tendency to overuse it, when it comes to generic products, I think that's an extremely appropriate description and aspect of our work in supporting advances in this industry. Certainly, the impact and benefits of generic products and the competition they produce to help lower costs and increase accessibility for patients cannot be overstated. According to the Association for Accessible Medicines, in 2021, the healthcare system saved $366 billion thanks to generic prescription medicines. Americans clearly appreciate and take advantage of these savings. Generics today represent 91% of prescriptions filled, accounting for 18% of all prescription drug spending. The value that patients place on generics is for good reason. They provide access to affordable, safe, effective, and high-quality medicines. This enables patients to obtain effective treatment at a low cost, clearly a public health priority for the FDA. We're taking the numerous actions to support the generics program. For example, OGD has continued its implementation of the FDA's Drug Competition Action Plan, or DCAP. Last October, we published draft guidances and product-specific guidances that provided recommendations for physicochemical and structural characterization tests, in vitro release test studies, and in vitro permeation test studies for topical generic drug products. These guidances will be discussed in today's workshop. One of the key aspects of our work to promote innovation in the generic drug space is our engagement with developers. Early communications can help reduce a generic drug product's time in the pipeline from concept to development to market input by strengthening development programs, improving FDA assessment efficiency, and enabling better ANDA submissions. In FY 2023, the FDA held numerous additional scientific meetings with industry as part of the first year of the second reauthorization of the Generic Drug User Fee Amendments, or GDUFA 3. Many of these meetings were related to complex products, which are harder to develop, and so those communications can reduce time and increase efficiency in the development process. Those meetings occurred before ANDA submission, during ANDA review, as well as after a complete response action. GDUFA 3 also included new goal dates added for publishing product-specific guidances, or PSGs, for newly approved complex products. Industry can also request to meet with FDA if in vivo bioequivalence studies have been started that are different from FDA's recently published new or revised PSGs. Since I mentioned GDUFA, let me focus on one of the most important components of GDUFA since it began more than 10 years ago. That is its emphasis on science and research, which is in line with the FDA's focus on and application of the best available science to help ensure the best technology, most definitive studies with clear results, and the best decisions. While the innovator deal with American society with patent protection for a limited period of time has undoubtedly stimulated drug discovery and new treatments, the generic deal is also critical, and science doesn't stop at the point of transition to generic status. The GDUFA Science and Research Program is designed to help enable the FDA to address particularly challenging scientific issues for product-specific guidance development and to communicate with applicants through pre-ANDA meetings to help clarify regulatory expectations for prospective applicants early in the generic drug development cycle. Let me briefly highlight one recently approved generic product that I think exemplifies the meaning of FDA-supported scientific innovation. and what can come out of years of GDUFA-supported research. In July 2023, the FDA approved the first generic to Vivitrol. Vivitrol is a 1-month extended release injectable naltrexone that is used to help people maintain abstinence while recovering from opioid or alcohol dependence. These and other naltrexone drug products are important tools to treat substance abuse. This is also the first generic that uses biodegradable PLGA, polymer microsphere technology. The PLGA microsphere formulation gives rise to the month-long extended release of naltrexone. Although a more complex product to develop than a once-daily tablet, the once-a-month extended release injection reduces the dosage regimen frequency and therefore may be advantageous in helping ensure patient adherence to the therapeutic regimen, which is critical to reducing the risk of relapse. The complexity of developing and manufacturing products that use control-release PLGA technology is exemplified by the limited number of products that have been approved, even though PLGA is a well-known and highly tunable material to create extended release formulations. Since the first FDA-approved PLGA product more than 30 years ago, there have been approximately 12 other approved products, and this is the first generic product that has PLGA. As with all companies developing a PLGA product, this generic had to address several complex challenges in developing, manufacturing, and demonstrating their naltrexone microspheres product is therapeutically equivalent to Vivitrol. OGD initiated its first research activities on PLGA-based long-acting products in 2013. The two main foci of these research activities were to develop analytical methods that can facilitate reverse engineering, characterization, and selection of PLGA, which is a critical first step for generic development. In vitro drug release testing methods was the second step to improve our understanding of how the drug is released from the formulation and how different polymer and manufacturing-induced characteristics affect drug release behavior. These methods have been included in FDA's product-specific guidance and have been adopted by a generic industry for supporting their generic development. In addition, the research on polymer and formulation characterization also enabled FDA to develop assessment criteria to make the generic pathway feasible for PLGA-based products. The approval of the first generic Vivitrol provides a good model for future generic development of PLGA-based products. GDUFA research will continue to explore how modern analytical methods and modeling tools can be helpful for guiding product development and bioequivalence study design. The improved understanding of the design space for this group of products will provide a good scientific foundation to explore novel approaches for product development and bioequivalence testing. In today's workshop, you'll have the opportunity to take a deep dive into some of the many ways that we work to modernize generic drug product development and enhance our science, policy, and business processes in support of this goal. In addition to strengthening our process to increase interactions with industry, to make submissions more complete and reviews more efficient, these modernization efforts include developing efficient bioequivalence approaches for products that eliminate unnecessary human studies and provide scientific approaches for generics for complex products, and helping industry maximize the use of the 505J pathway and gain clarity around how to develop generic versions of complex products, such as generic drug-device combination products. Before I close, I want to mention one other important facet of generic drug development, which is its global significance and impact. As we all know, the development of drugs today, including generic drugs, is part of a global enterprise and the product of a global supply chain produced with the intent to market in multiple countries around the world. I'm pleased to see that one of today's sessions will focus on the topic of global collaboration to support efficient generic product development and regulatory assessment. Though it's not the primary focus of the discussion, I think it's important to mention one aspect of this dynamic, which is the challenge of drug shortages. Shortages are very frustrating, consume an enormous share of talented pharmacist and clinician time to find alternatives and explain these issues to patients, undermine the underlying goal of increased accessibility of these often life-saving products, and indeed, simply put, shortages put patients at risk. As you know, the FDA doesn't make drugs, and we can't tell manufacturers which or how much of a drug to produce. But we do try to anticipate shortages and take action to prevent them, including working with drug makers to submit applications for new manufacturing capacity to increase supply of existing drugs or to facilitate market entry for new drugs. In addition, we've encouraged the adoption of advanced manufacturing technologies and mature quality management practices to help avoid or mitigate drug shortages. We also partner across government, academia, health systems, and industry to strengthen and diversify the supply chain and identify and address supply chain vulnerabilities and accelerate domestic manufacturing. There are many reasons for drug shortages, but one I want to mention involves manufacturers of older generic drugs, and particularly injectables, who sometimes face intense price competition, uncertain revenue streams, and investment requirements to maintain quality conditions. U.S. patients benefit from access to these less expensive generic drugs, but if the basic economics and contracting practices of the generic drug market are not fixed, more patients will be impacted by these shortages. We need to develop transparent measures of the quality of manufacturing coupled with contracting practices for high-quality manufacturers that ensure the supply of a high-quality product. This is just one of many topics you'll be addressing in your discussions today. The diversity of generic products is enormous, as the meeting agenda makes abundantly clear. You'll have discussions related to complex APIs, complex injectable products, topical drug products, ophthalmic emulsion products, orally inhaled drug products, noteworthy guidance development and updates, new processes, research and assessment tools, common issues related to the ANDA process, and more. Quite a meeting. In short, you've got a full plate, and we're here to support both the discussion and the developments that we hope flow from them. By working in mutually supportive ways, we can translate science to approval and continue to deliver timely, high-quality, safe, affordable, and effective generic products to the public. Thank you again for your participation, and I hope you have an engaging and productive day.
Thank you for that great keynote address. This is Ray Ford working with SBIA to serve as your moderator and host for today's remaining sessions. Welcome to session 1, Noteworthy Guidances and Generic Drug Approvals for Topical and Transdermal Products. Our speakers for this session include Dr. Priyanka Ghosh, Lead Pharmacologist, Division of Therapeutic Performance 1, Office of Research and Standards in OGD, and Dr. Hiran Patel, Senior Staff Fellow, Division of Bioequivalence 2, Office of Bioequivalence in OGD. Next, we'll hear from Dr. Megan Kelchin. Dr. Kelchin is a senior Pharmacologist, Division of Therapeutic Performance 1 in the Office of Research and Standards OGD. And finally, before our break, Dr. Ahmed Zidan will present. Dr. Zidan is a Senior Staff Fellow in the Division of Product Quality Research, Office of Testing and Research in the Office of Pharmaceutical Quality. For a complete bio on each presenter today and Thursday, please refer to our speaker's biography located in the same area on our website as the conference agenda. Now, please join me to welcome our first presenter, Dr. Ghosh.
Good morning, everyone. Thank you so much for the kind introduction. My name is Priyanka Ghosh, and this morning I'm going to talk about the general guidances that we have published in 2022 related to characterization-based bioequivalence approaches for topical products. In part 1 of this talk, I'm going to talk about the Q3 guidance. In part 2, Dr. Hiren Patel will cover the IVRT and the IVPT guidances. This presentation reflects my views and should not be construed to represent FDA's views or policies. The learning objectives for us this morning are to understand the key concepts in the general guidances related to Q3 sameness versus Q3 similarity, and understand how an evaluation of formulation sameness or the no significant difference standard influences categorization of a product as Q3 same or Q3 similar. The guidance I'm going to talk about today is physicochemical and structural characterization of topical drug products submitted in ANDAs. We often refer to this as Q3 guidance in short. And towards the end of the talk, I'm going to talk about how to interact with the FDA if you are interested in developing a Q3 similar product. Before I go into the definition of Q3, I briefly want to remind you about Q1 is the components in the drug product. These are the inactive ingredients that make up the drug product. Q2 is the composition of the product. These are the concentration of each of the inactive ingredients in the drug product. One key thing to remember are topical products applied to the skin are not required to be Q1, Q2 the same as the reference listed drug by regulation. However, we often recommend Q1, Q2 sameness or formulation sameness in our guidances. When we do recommend formulation sameness in our guidances, we state that the test product should contain no difference in inactive ingredients or in other aspects of the formulation related to the reference standard, or in short, the no significant difference standard. Now, while I will not be able to go into the details of the similarities or differences between a Q1, Q2 assessment and a no significant difference assessment within the scope of this talk today, I encourage you to review this presentation from November of 2022, which outlines the agency's current thinking related to the no significant difference assessment. Now that we have reviewed Q1 and Q2, let's talk about Q3. Q3 is the arrangement of the active and the inactive ingredients in a product. This is an understanding of the microstructure of the drug product. And when we talk about comparative Q3 characterization in our guidances, what we are really referring to is measuring specific Q3 attributes such as the pH, the particle size, the globule size, and comparative assessment of each of those characteristics of the prospective test product and the reference product. Now, this is just an example from our research, and I included this to illustrate that when we typically recommend characterizing 3 batches of the test product and 3 batches as available of the reference product for comparative Q3 characterization. And for a product to be categorized as having the same arrangement of matter, the expectation typically is that the observed data for the test product will fall within the variability of the observed data for the reference product. So again, the expectation is that the data that we obtain for the test product falls within the variability of that obtained for the reference product. And this is important to remember, because the Q3 guidance talks about categorization of products as Q3 same, Q3 similar, and Q3 different. So how do we do that? In the guidance, the first question we ask is the arrangement of matter the same? If the arrangement of matter is the same, then the second question we ask is the underlying matter the same? What this really means is, does your product meet the no significant difference criteria? If the answer is yes, we categorize those products as Q3 same, and we have recommendations for products that meet the Q3 sameness criteria in our product-specific guidances. On the other hand, if the arrangement of matter is not the same, we categorize these products as Q3 similar. At this time, we don't have recommendations for these types of products in our guidances, and therefore we strongly encourage you to come meet with us through the pre-NDA program if you intend to develop a product, a Q3 similar product. Lastly, if the arrangement of matter is not the same, those products are categorized as Q3 different products. And for these products, we often recommend either using the comparative clinical endpoint bioequivalence studies or the vasoconstrictor studies when those are relevant for establishing BE. So now that we have talked about Q3 sameness, Q3 similar, Q3 difference, let's discuss a little bit about what are the specific Q3 characteristics that we typically recommend in the general guidance and in our product-specific guidances. The Q3 guidance summarizes basic Q3 characterization. These are characterization of appearance and texture, the phase states, and the structural organization of matter. And what we really mean by this is, for example, does your product have globules? Does your product have particles? And this information really allows us to assess if your prospective test product is the same dosage form as the reference product, and therefore supports an evaluation of pharmaceutical equivalence. In our guidances, the product-specific guidances, we often recommend comprehensive Q3 characterization. And these include, in addition to the basic Q3 characterization, characterization of polymorphic form, rheological behavior, water activity, drying rate, pH, etc. And the types of Q3 characterization that we identify and recommend in a product-specific guidance for a given reference standard depends on an understanding of the microstructure of that reference product itself. And Dr. Kelchen, in her talk a little later this morning, will demonstrate how we identify some of the relevant Q3 characteristics that we recommend for a specific reference product in our guidances. So with that background in mind about Q3 characteristics and Q3 sameness versus similarity versus difference, let's talk a little bit about how to interact with the FDA if you are interested in developing either a Q3 same or a Q3 similar product. So if you are interested in developing a product that meets the no significant difference criteria, which is essentially a product that may be characterized as a Q3 same product, if a product-specific guidance is available, and we have about 80 of those currently published. If you have a question, you can always submit a control correspondence. If you would like to propose an alternative approach, you can always submit a pre-NDA meeting. If a PSG is not available, we encourage you to review the relevant general guidances, and we encourage you to submit a pre-NDA meeting, including your proposed formulation. You can include up to 3 formulations in a given pre-NDA for assessment, and any empirical data you may have to support your proposed bioequivalence approach. On the other hand, if your product does not meet the no significant difference criteria, or products that may be categorized as Q3 similar products, we encourage you to review the relevant general guidances, but we strongly encourage you to come and talk to us using the pre-NDA program. And for these kinds of products, we want you to summarize your assessment of what the differences in formulation are between the proposed test product and RS, any Q3 characterization data you may have that illustrates why you think your product meets the Q3 similar criteria, and finally includes your proposal for the types of studies you would like to conduct to mitigate the risk associated with potential failure modes for BE that may arise due to the differences in formulation. And when you include studies, we really prefer that you include any preliminary data you may have, because that really helps us to assess the viability of your proposed approach. So with that in mind, let's go to a challenge question. My challenge question for you this morning is, which of the following statements is true? Basic Q3 characterizations can demonstrate that a test topical product and its RS are the same dosage form, supporting a demonstration of bioequivalence. Comprehensive Q3 characterizations matching the detailed profile of the Q3 attributes of the test product to the detailed profile of the Q3 attributes of the RS substantially mitigates the risk of potential failure modes for bioequivalence, supporting a demonstration of bioequivalence, and all of the above. The correct answer is B. Basic Q3 characterizations typically support a demonstration of pharmaceutical equivalence, and therefore this statement is not entirely true. With that, my acknowledgements for our entire team at the US FDA, specifically Dr. Sam Rainey, Dr. Markham Luke, Dr. Paula Simamora, and Dr. Bing Kai, who were heavily involved in the development of the Q3 guidance. And I would also like to thank all of you for for attending this talk this morning, and I look forward to your questions during the panel discussion. Thank you.
Good morning, everyone. Thank you for the nice introduction, and thank you, Dr. Ghosh, for a very informative presentation to kick off this session. Now I'm going to continue with two more guidances for characterization-based bioequivalence approaches for topical products. Before going into the details of my presentation, I have the following disclaimer. This presentation reflects my views and should not be construed to represent FDA's views or policies. The learning objective for my today's talks are to discuss general recommendations pertaining to critical aspects related to conduct and analysis of in vitro release stage, that is IVRT, and in vitro permeation test studies, that is IVPT, for submission in ANDAs for topical drug products. Also, to identify important updates provided in the scientific recommendations within the general guidance for IVRT and IVPT studies. Let's begin with in vitro release stage, that is IVRT. As you all may be aware that the agency laid out the recommendation for the conduct and analysis of IVRT studies in the draft PSG of acyclovir cream before October 2022. The recently published new general guidance on IVRT in October 2022 now reflects the current thinking related to the IVRT studies for topical products submitted in ANDAs. For today's discussion, I'll be focusing on the important updates in terms of involvement in the scientific recommendations within the IVRT guidance. I would suggest you to go to the detailed recommendation in the IVRT general guidance before you plan on conducting IVRT studies. Let's begin with IVRT method development. The guidance provides in-depth recommendations for the selection of critical IVRT method parameters, including receptor solution and membrane, as a part of method development. As you may know that the USP General Chapter 1724 historically recommends the typical IVRT study duration to be between 4 and 6 hours. In multiple instances, the shorter IVRT study duration was used without adequate justification. Therefore, the guidance emphasized that the study duration should be at least 4 hours to capture the release rate which can represent the steady state release. Under very limited circumstances with adequate justification, also supported by data, may be considered acceptable, but it is not typically recommended. The linearity of the drug release rate, or the slope, across all time points should ideally have an R-square value of greater than or equal to 0.97, which was previously 0.9 in the draft PSG of acyclovir cream. Let's move on to the IVRT method validation. The guidance provides recommendation on what type of data can support membrane surface temperature as a part of equipment qualification. The equipment was termed as apparatus previously. Membrane inertness should be evaluated in relation to the membrane binding of the drug in the receptor solution at a concentration relevant to the range of drug concentration in the receptor solution during the test. To add to this, one concentration toward the lower end and one on the higher end of the study sample should support the range of drug concentrations. The guidance further provides details on expectation for receptor solution sampling qualification. For precision and reproducibility, it is expected to have a minimum of 3 independent runs with the nominal strand to support this particular validation parameter. The linearity of drug release across all time points should be calculated and reported for each diffusion cell and compared within and across all IVRT runs. As I mentioned earlier, for the release rate to be considered suitably linear, it should have an R-square value greater than or equal to 0.97 across IVRT study duration. Moving forward, it is recommended to report dose depletion only and not necessarily mass balance and recovery. In this regard, the guidance further indicates that the IVRT method may be considered adequate despite a depletion of greater than 30% when experimental evidence illustrates that the release rate or slope remains suitably linear for each diffusion cell when plotted versus the square root of time. I would like to make a note that only the explanation on dose depletion higher than 30% may not be appropriate to have a shorter study duration. Next validation parameter is sensitivity, specificity, and selectivity. It is typically recommended to compare the release rates from the test formulation with the higher strength, that is 150%, and a lower strength formulation, that is 50% of the nominal strength. There may be challenges under certain situations. For example, if precipitation of the active ingredient is observed when formulating the 150% strength, it may be necessary to use different strategies which may be discussed with the agency before the submission of ANDA. For IVRT specificity, an IVRT method should be proportionally linear for the correlation of the formulation concentration to the average IVRT release rate with a minimum R-square value greater than or equal to 0.95, which was 0.9 previously. To support IVRT selectivity, both alter formulation, that is 50% and 150% of the nominal strength, should fail to show equivalence to the release rate from the nominal strength formulation using statistical approach. The supplemental demonstration of the selectivity of the IVRT method further validates that it can detect the difference in the release rate which are associated with aspect of the formulation other than the strength which was demonstrated under fundamental selectivity. The alter formulation may utilize different strategies which include but not limited to the change in an active ingredient and or its concentrations, the manufacturing process, or combination of this. In addition to this important aspect, the guidance also provides some additional detailed recommendations. Like there is a separate section about sample analytical method validation, handling and retention of sample, control of study procedure with quality management system, as well as how and where to submit the specific study information in ANDAs. In the interest of time, I will not go in details as it is self-explanatory. With that, it's time for the first challenge question. If the dose depletion is higher than 30% for the IVRT study, despite the linearity of IVRT method greater than 0.99 for all cells, the IVRT method is not considered acceptable. Is it A, true, or B, false? If you selected the option B, false, then you are correct. Time to move to the next general guidance for the in vitro permeation test, that is IVPT. As I mentioned earlier, the new general guidance on IVPT reflects the current thinking related to the IVPT studies for topical drug products submitted in ANDAs, and it supersedes the recommendation in the draft PSU of acyclovir cream. Similar to the IVRT guidance, I'll be focusing on the important updates in terms of evolvement in the scientific recommendation within the IVPT guidance. Let's begin with IVPT method development. The guidance provides in-depth recommendation for the selection of critical IVPT method parameters, which include but not limited to equipment, termed as apparatus previously, dose amount, skin source, type and preparation, along with its barrier integrity test, the study duration, and the dose depletion if the wiping off procedure has been utilized. I will not go in details for each parameter in this slide. Once these preliminary parameters are laid out, it is critical to optimize the dose amount in terms of the consistency with which the dose can be applied. the reproducibility of the flux profiles, the influence of dose amount and dose duration on the shape of the flux profile, and the approximate range of drug concentration in receptor solution samples at different time points. Parallelly, the sampling schedule and study durations should also be optimized that ideally should include a complete flux profile to identify the maximum flux and a decline in the flux thereafter across multiple subsequent time points. In certain situations, the decline in the flux may not be observed, for which the alternate approach may be used. The guidance provides recommendation to utilize wiping off the applied dose after a suitable duration from the skin and continue to monitor the receptor solution for an extended period. The next parameter is the skin barrier integrity testing. Previously, the most common types of testing were listed. For example, transepidermal water loss, that is TWL, tritiated water, electrical base, for example, transepidermal electrical resistance. However, new guidance provides detailed recommendations about the process for each type of testing along with considerations on acceptance criteria. I would like to make a note that the testing data may not correlate with the permeation of most topically applied active ingredients, and it also recommends the do's and don'ts while conducting respective testing. Now let's move on to the IVPT method validation. I'll begin with the receptor solution qualification. Similar to the previous recommendations, the composition and pH of the receptor solution should be qualified in relation to the stability and solubility of active moiety in the receptor solution, along with the use of antimicrobial agents such as around 0.1% sodium azide or around 0.01% gentamicin sulfate to mitigate potential bacterial decomposition of the dermis and epidermis. For hydrophobic drugs, the solubility of active moiety is always a challenge in the physiological buffer-based aqueous receptor solution. It may be appropriate to use 0.1 to 0.2% polyoxyethylene ole ether in the receptor solution to enhance solubility of hydrophobic drugs. I would like to make a note that the use of organic solvents and alcohol in the receptor solution to enhance solubility of active moiety are not recommended, as it may have potential to alter the skin barrier function and ultimately alter the permeability of the skin. Let's move on to the foundation of IVPT method validation. The discrimination ability of the IVPT method may be described using two concepts, sensitivity and selectivity. The IVPT sensitivity studies are typically performed towards the end of the IVPT method development phase, and the key purpose of this study is to incorporate the final IVPT method parameters like target dose duration, dose amount, and study duration to be used in the IVPT method validation or pilot and pivotal study. In terms of IVPT selectivity or pilot study, it should be performed once the IVPT method parameters are established. The IVPT pilot study typically supports multiple IVPT method validation parameters. I would like to make a note that IVPT pilot and pivotal study parameter should be the same as the IVPT sensitivity study. Let's look at the consideration individually, and we'll begin with IVPT sensitivity. While the draft PSC of acyclovir cream enlisted modulation of dose amount, dose duration, and product strength approaches to demonstrate IVPT sensitivity, the new guidance provides detailed recommendations or reflect current thinking for each approach. For example, in terms of modulation of dose amount, it recommends the selection of lower and higher dose amount that is threefold compared to target dose amount as a starting point. Also, it further provides scientific considerations for the dosage form and formulation along with its limitation for the utility of this approach. For the modulation of dose duration, the guidance recommends selection of lower and higher dose duration compared to target dose duration using the controlled dose amount across all treatments along with preliminary thinking on which type of drug product this approach is more suitable. In terms of modulation of product strength, the guidance indicates that the utility of this approach should be considered only under certain situations because of the overall limitations. Moving on to the IVPT pilot study, which should be conducted with 3 parallel treatment, taste, reference, and a topical product or formulation that is known or designed to be different from the reference topical products using multiple donors, for example 4 to 6, with a minimum 4 replicates per donor per treatment. The pilot study supports a validation of IVPT method parameters like permeation, profile and range, precision and reproducibility, dose depletion, and IVPT selectivity. This data further help in estimation of number of donors that may be needed to adequately power the pivotal IVPT study. Speaking of the pivotal IVPT study, the IVPT endpoints include maximum flux, that is Jmax, and total cumulative amount permeated into the receptor solution across the study duration, that is AMT. Please note that this was termed as AUC in the draft PhD of acyclovir cream. The flux profiles should be plotted as the flux on the y-axis versus time on the x-axis, and the maximum values for each diffusion cell provides Jmax. The extent of drug permeation should be plotted as the total cumulative amount of drug permeated on the y-axis versus the time on the x-axis. Let's talk about the statistical analysis. If SWR value of the reference product is greater than or equal to 0.294, it is recommended to use reference scaled average bioequivalence approach, otherwise use the average bioequivalence approach for B determination. At the completion of the study, if the number of skin replicates are the same for all donors in the taste and reference topical product treatment groups in the IVPT study, use a balanced design statistical analysis. If the skin section or diffusion cells are excluded from the final statistical analysis because of experimental loss or issue and the resulting dataset is unbalanced, then use an unbalanced design statistical analysis. I would like to make a note that the study design should always be balanced despite of the data being balanced or unbalanced. In addition to this important aspect, the guidance also provides some additional detailed recommendations. Like there is a separate section about sample analytical method validation, handling and retention of sample, control of study procedure with quality management system, as well as how and where to submit the specific study information in ANDAs. In the interest of time, I will not go in the detail as it is self-explanatory. Time for next challenge question. To support IVPT sensitivity using modulation of dose duration approach, the dose amount should be the same across different dose duration, that is low, target, and higher dose duration. Is it A, true, or B, false? I'm going to give you 10 seconds to think about this one. If you selected the option A, true, then you are correct. I have added some resources in the slide which maybe add further value for your topical drug product development program using characterization-based approaches. With this, I would like to acknowledge my OB colleagues, Drs. Nair, Dr. Vivian, Dr. Chiang, Dr. Chang, Dr. Lee, Dr. Roy, as well as my ORS colleagues, Dr. Ramazanli, Dr. Ghosh, Dr. Rane, and bioequivalence assessors. Last but not the least, I would like to thank all of you to patiently listen to my presentation. Please keep sending your questions and I'll be happy to address as many questions as we can during the Q&A session. Thank you.
Hello, my name is Megan Kelchin, and today I'll be providing an overview of the current product-specific guidances for topical products that are applied to the skin. Here is my disclaimer. After today's presentation, you'll be able to identify the various approaches for bioequivalence for topical products that are outlined in product-specific guidances, or PSGs. You'll also be able to explain the rationale for inclusion of different components within the scope of a characterization-based bioequivalence approach for topical products, and describe how to obtain the agency's feedback on a proposed bioequivalence approach when a PSG is not available. So let's first go over how we might demonstrate bioequivalence for a topical product. The first approach is a comparative clinical endpoint bioequivalence study. This is an in vivo study in which the test product is shown to be as efficacious as the RLD, and both products are shown to be superior to a placebo control. The next approach is a characterization-based bioequivalence approach. This is a weight of evidence approach comparing the formulation, microstructure, and performance of the test product and the RLD. And I'll discuss the characterization-based bioequivalence approach in more detail in the coming slides. Next is an vasoconstrictor or VC study, and this is an in vivo study comparing the skin blanching effect of the test product and the RLD. Additionally, we may recommend a waiver of in vivo bioequivalence studies for some products, and to support the request for a waiver, we may compare the formulation or the dosage form of the test product. It's important to note that within a single PSG, there may be multiple bioequivalence approaches. For example, a single PSG may contain two options, a characterization-based approach or a clinical endpoint study. And another example would be a PSG that recommends a waiver of in vivo BE studies or a VC study if a test product does not meet the requirements of the waiver. Topical products are utilized for a variety of indications, and it's these indications that we recommend as part of our comparative clinical endpoint studies. However, it's important to keep in mind that a single indication may have multiple ways of establishing bioequivalence, and this is dependent on things like the active ingredient, the dosage form, or aspects related to the disease. So let's look at psoriasis as an example of this. So shown in the graph are the different types of drugs that are indicated for the treatment of psoriasis. And if we look at the vitamin D derivatives, we see that there are three types of bioequivalence approaches that we recommend in our PSGs for these products. A waiver of in vivo BE studies may be recommended for solution-based products that meet a formulation sameness standard compared to the RLD product. For semi-solid drug products, we may recommend a characterization-based bioequivalence approach for a test product that meets a formulation sameness standard compared to the RLD. And for products that do not meet the formulation sameness standard of the waiver or the characterization-based approach, we recommend include a recommendation for a comparative clinical endpoint BE study. For corticosteroid products, we recommend similar types of bioequivalence approaches. For example, we recommend a waiver of in vivo studies or a characterization-based bioequivalence approach when the test product meets a formulation sameness standard. However, because corticosteroids have a skin blanching effect, we commonly recommend a VC study for these products. We do not recommend a VC study for products intended to be applied to the scalp, so in those situations, we recommend a comparative clinical endpoint study for products that do not qualify for the waiver approach or the characterization-based approach. For products that contain both a corticosteroid and a vitamin D3 derivative, we can recommend a combination of bioequivalence approaches. For example, we might recommend a VC study in addition to a comparative clinical endpoint study, or VC study in addition to a characterization-based bioequivalence approach. So the take-home message for this slide is that there are often multiple approaches for establishing bioequivalence for topical products, even within a single indication, and these recommended BE approaches within PSGs are product-specific. As Dr. Ghosh and Dr. Patel mentioned in the previous presentation, in October of 2022, the agency published 3 new draft guidances for industry related to Q3 characterization, in vitro release tests, and in vitro permeation tests for topical products. Additionally, the agency published a revised draft guidance for industry related to in vivo bioequivalence for topical corticosteroid products, and this focuses on those VC studies. This guidance will be discussed in the second part of this session today. In addition to the general guidances, we also published approximately 80 new or revised PSGs for topical products. The majority of these PSGs included characterization-based bioequivalence approaches that aligned with the recommendations in the new general guidances. So let's talk about the components of these characterization-based approaches. The first component is the no significant difference standard. The no significant difference standard is a comparison of the test product formulation and the RLD formulation, and is based upon the principles of assessing Q1, Q2 sameness, but also considers certain differences that have previously been determined by the agency to be acceptable based on available scientific evidence. And it's important to note that a no significant difference standard expands the eligibility for a characterization-based approach for topical products. So as an example of the no significant difference standard is a test product that contains a minor differences in inactive ingredients. For example, an RS formulation may use carbapol 934P, which is a benzene-containing carbomer that is not preferred by the agency. Under the no significant difference standard, a test product may use a carbomer homopolymer type B, which is non-benzene containing. I refer you to the December 2022 FDA CRCG presentation that's highlighted here if you'd like to learn more about the no significant difference standard. The next component is a physical, chemical, and structural characterization, or comparative Q3 characterization. And Q3 characterization is recommended to mitigate failure modes for bioequivalence associated with differences between the test product and the reference product in the microstructure. And you'll note here that in the snapshot of the PSG that's shown here, there are a variety of different Q3 attributes that are recommended. And the recommended Q3 attributes for a specific product are based on an understanding of the microstructure of that product. The next component is an in vitro release test, or an IVRT study. An IVRT study is a very sensitive and reproducible test that's able to capture differences between the test product and the RLD that may not have been captured with formulation sameness or Q3 characterization. For more complex dosage forms, we often recommend an in vitro permeation test or an IVPT study. This study is recommended to mitigate failure modes for bioequivalence associated with complex interactions between the formulation and the skin. For example, an IVPT study may be recommended for drug products that are multiphasic in nature or contain complex and active ingredients. An example of this would be petrolatum-based ointments. Finally, in some cases, an in vivo systemic PK study may be recommended when there's a question of whether the systemic drug concentrations contribute to the efficacy of the drug product. So as you can see on the graph here, we have developed characterization-based approaches for a variety of dosage forms, and I'll talk about this more in the coming slides. You'll also notice that at this time, characterization-based approaches are included in only a subset of published PSGs. I wanted to note that at this time we are actively working on incorporating characterization-based approaches into both new and existing PSGs. However, if you would like to propose a characterization-based approach as an alternative BE approach to what is recommended in the PSG, we encourage you to submit your proposed BE approach through the appropriate communication pathway, such as a pre-ANDA meeting request. And I'll discuss the recommendations for the pre-ANDA meeting request process later in my presentation. But for now, let's look at the components of the characterization-based approach, starting with the topical aqueous gels. First, I'll note that a no significant difference standard is recommended for all characterization-based approaches within PSGs at this time. And that's because at this time, Q3 sameness is recommended within the scope of the PSGs, and that relies on a test product that meets the no significant difference standard. Next, all characterization-based approaches within PSGs include a recommendation for basic Q3 characterization. And this includes an assessment of visual appearance and texture and microscopic images. And this is to understand the structural organization of matter of the test product and the RLD. The rest of the Q3 are recommended based on the microstructure of the drug product. So in the case of topical aqueous gels, we always recommend rheology, pH, and specific gravity. Particle size distribution is recommended when there's suspended drug in the finished drug product. The majority of topical gels are single-phase drug products, and so for these products, we do not recommend an assessment of globule size distribution. And finally, for topical aqueous gels, these products typically contain large concentrations of water, so Q3 attributes such as water activity, drying rate, and oleaginous components are not considered to be relevant for these drug products. I'll note that an IVRT study is recommended as part of a characterization-based approach for all drug products, And again, this is to capture differences between the test product and the RLD that may not have been captured with formulation sameness in Q3 characterization. Finally, I'll note that you'll notice that for diclofenac sodium topical gel, this drug product is multiphasic. So for this product, we recommend an assessment of globule size distribution in addition to a recommendation for an IVPT study. Additionally, it's suggested that this drug product has a systemic contribution to efficacy. So in this case, we recommend a systemic PK study. So how do these recommendations compare to the recommendations for topical non-aqueous gels? Well, you'll note that for the non-aqueous gels, there's typically a high concentration of volatile and active ingredients. And so for these products, we recommend an assessment of drying rate. Additionally, these non-aqueous gels often contain low concentrations of water, and so an assessment of pH is not recommended for every product like we see for the topical aqueous gels. I'll also note that all of the non-aqueous gels are considered to be single phase, and so we do not recommend an assessment of globule size distribution, nor do we recommend an IVPT study. So let's move on to a little bit more complex drug product like topical creams. So you'll note that the majority of topical creams are multiphasic, and this is evident by the recommendation for globule size distribution for almost every topical cream product. And for these products that are multiphasic, we include a recommendation for an assessment of IVPT as part of the characterization-based approach. However, you'll note that there are some exceptions to this rule, as there are some PSGs that are multiphasic and contain the recommendation for globule size distribution, but do not contain an IVPT study. And there are specific situations in which we might not recommend an IVPT study as part of a characterization-based approach. So for example, for mupirocin calcium and silver sulfadiazine topical creams, these drug products are applied to the skin with no stratum corneum. And so therefore, we do not require an assessment of the interaction between the formulation and the skin via an IVPT study. For dicosanol is considered to have unique physicochemical properties and dosage form, and therefore does not necessitate an IVPT study. Finally, some drug products have a site of action that is in the superficial stratum corneum. These products might include things like ketoconazole and butanafine hydrochloride topical cream, which are indicated for treatment of fungal infections where the infection is located in the superficial stratum corneum, and therefore an IVPT study is not needed. I'll note that when an IVPT is not recommended as part of a characterization-based approach for a drug product that is multiphasic in nature, we do recommend an assessment of drying rate for those products. And that's to evaluate differences in the metamorphosis of the drug product over time, which would typically be captured via an IVPT study. Like the topical creams, topical lotions are mostly considered to be multiphasic drug products, as you can see by the recommendation of globule size distribution for all lotion products. Similar to the creams, though, you'll see that an IVPT study is recommended for most topical lotions, but there are some notable exceptions. For example, in the case of ammonium lactate, the site of action is in the superficial stratum corneum. And then for this product, we recommend an assessment of drying rate to capture those differences in metamorphosis. In the case of benzyl alcohol or ivermectin, these drug products are indicated for the treatment of lice, and the site of action is an external organism, in this case, the lice on top of the skin. So for these products, we recommend an ex vivo pediculicide hair tuft assay as the biorelevance study for these drug products in lieu of the IVPT study. So as previously mentioned, an IVPT study is commonly recommended when the drug product is multiphasic or if it contains complex and active ingredients such as a large concentration of oleaginous components. So if we look at the topical ointments, you'll see that when a PSG recommends both an assessment of oleaginous components and assessment of globule size distribution, we also recommend an IVPT study. Even for products where the drug product does not contain globules but does contain a large component of or a large concentration of oleaginous components, we recommend an IVPT study. However, as always, there is a notable exception. For miconazole nitrate, white petrolatum, and zinc oxide, the drug product contains a large large concentration of oleaginous components, but does not include an IVPT study, and this is due to the indication of the drug product. Finally, I want to highlight that the recommendations within the PSGs are research-driven, and a great example of this is Transdermal topical ointment. Dr. Zaiden will be discussing the research that was done to support the development of this specific PSG in the next presentation. So you might be thinking, what happens if I want to develop a generic for a specific product that does not have a PSG? The first thing I recommend is to check the FDA's upcoming PSG website. This contains a list of PSGs that OGD plans to publish within the next 12 months. You can also send in a request for guidance development, and this helps OGD identify which products are of high interest to the agency. Finally, you can submit a pre-ANDA product development meeting request with your proposed bioequivalence approach. In a pre-ANDA meeting request, we recommend you include the following information: a clear, specific, and comprehensive outline of your exact BE approach, specific questions related to your proposed BE approach or your product development program, a formulation table with up to 3 proposed test formulations, and empirical data to support the proposed BE approach. So let's move on to some challenge questions. Challenge question number 1, which of the following is false? A, when a PSG contains a characterization-based approach, a no significant difference standard is always recommended. B, comparative Q3 characterization is always recommended. C, an IVRT study is always recommended. Or D, a systemic PK study is always recommended. The answer is D. A systemic PK study is not always recommended as part of a characterization-based approach for topical products. Challenge question number 2. In general, when is an IVPT study recommended as part of a characterization-based approach for topical products? A, when the drug product is multiphasic. B, when the drug product is a petrolatum-based ointment. C, always. Or D, A and B, when a drug product is multiphasic or when the drug product is petrolatum-based. The answer is D. So in summary, PSGs for topical products contain harmonized recommendations in alignment with the relevant general guidances. The recommendations within a characterization-based bioequivalence approach for a topical product are product-specific and are based on an understanding of both the microstructure of the drug product and the performance of the drug product. Finally, when a PSG is unavailable, applicants are encouraged to submit a pre-ANDA product development meeting request to get the agency's feedback on their proposed bioequivalence approach. With that, I'd like to acknowledge all of my colleagues at the FDA. And I also would like to direct you to a number of resources that may be helpful as you start to develop your characterization-based bioequivalence approaches. With that, I'd like to thank you for your attention, and I look forward to answering your questions during the panel session. Thank you.
Good morning. In today's talk, we are going to discuss how research supports the development of product-specific guidance for topical drug product. I am Ahmad Zidan. I am a senior staff pharmacologist at DBQR of FDA. First, let me ground my presentation with this statement of pharmaceutical quality. Everyone deserves confidence in his next dose of medicine, and pharmaceutical quality, it assures the availability, safety, and efficacy of every dose of the medicine. Pharmaceutical research is not only the heart to develop and manufacture of topical drug product, but also it is hard to develop guidelines and to ensure that a science-based decision about drug approval is taken. Pharmaceutical research is involved in all stages of life cycle of drug product, starting from formulation development and selection of excipients and running compatibility studies, and in formulation design to ensure an enhanced topical penetration and delivery of the drug. It is involved in preclinical studies to get more information about in vitro performance, for example, the drug release rate and drug permeation across the skin, and to get more information about the rheology and sensory attribute of the formulation. And also it's involved in the in vivo studies to get more information about the pharmacokinetic biodistribution and elimination after topical application. It is involved in the safety and efficacy assessment, for example, to ensure safety of topical product, in development of skin irritation studies, and in clinical trials to get information about the clinical endpoint studies to ensure efficacy and dosing and patient outcome. And also it involved in the robust data collection and statistical analysis, and this is all to comply with the regulatory requirement. And the main outcome is to ensure that the generic product is similar to the reference drug product in safety and efficacy and also of quality. So in my talk today, we are going to cover how research contributed to availability of generic topical products and how research is used for demonstration of bioequivalence, in particular about in vitro characterization to support the product-specific guidance development. And we are going to highlight two of FDA case studies about the physicochemical research that supported BSG development. We are going to talk about terbinafine topical ointment and clascoterone topical creams. And we will have some challenging questions and key takeaways. So by the end of my presentation, the audience should gain a comprehensive understanding of the framework used to assess bioequivalence of generic topical product, and they should explore the multifaceted aspects of bioequivalence, including in vitro dissolution permeation study, quality equivalence evaluation, safety, efficacy assessment, and label consideration. And they should comprehend the pivotal role of pharmaceutical research in shaping the product-specific guidance for topical drug products. Indeed, a comprehensive research process is fundamental to ensure the availability and quality of generic topical product. This research facilitates overcoming the barriers for developing generic topical products in terms of demonstrating bioequivalence, ensuring formulation similarity, establishing IVIVCs, assessing sensory experience, and setting reference standards, and understanding absorption variability, and confirming therapeutic effectiveness, and conducting comparative clinical trials, and monitoring product post-market. So let's go over some of these points in detail. For example, bioequivalence studies are done to demonstrate that the generic product performs similarly to the reference product in terms of drug release and absorption. And these studies are essential to establish that the generic product is therapeutically equivalent to the innovator product. And the qualitative and quantitative variability and excipients between generic and reference product may impose a challenge to demonstrate formulation sameness and similarity, specifically because it impacts the drug release and absorption, making it more challenging to demonstrate bioequivalence. So establishing also the correlation between the in vitro dissolution testing and the in vivo performance parameter is crucial for development of generic topical product. And demonstrating that the in vitro performance matches the in vivo performance of the reference product, it may be a barrier for availability of generic topical products. And the patient sensory perception of generic product can also be influenced by factors like packaging, branding, past experience with generic medications. So negative perceptions may be challenging and may result in deterrence from trying a generic product, and it affects the availability of a generic topical product. And sometimes it's also challenging to determine what is the reference standard for a generic topical product, and lack of appropriate reference standard may hinder demonstration of comparability. Topical drug products such as creams and ointments can have a variable absorption rate due to difference in excipients or formulation design or structure. So achieving consistent and comparable absorption to a reference product may be challenging for development of generic. And the last but not the least is that ensuring the generic topical product have a same therapeutic effect as the reference product is essential for a regulatory approval and market acceptance. And any differences in therapeutic effectiveness may pose a barrier for availability of generic product. And this is where the research comes to alleviate these barriers and make generic topical products available to the patients. Demonstrating equivalence of topical product involve a single or a combination of the in vivo, in silico, and in vitro approaches. Historically, in vivo studies such as in vivo pharmacokinetic studies, in vivo pharmacodynamics such as vasoconstriction studies, and in vivo comparative clinical endpoint bioequivalence studies are used, which are comparative clinical trials conducted on human subjects to directly assess the clinical equivalence by monitoring absorption, therapeutic effectiveness, safety, and adverse effects. In silico modeling approaches are also used for demonstration of bioequivalence. For example, computer-based simulation predict how the drug behaves without the physical experiments. Examples include pharmacokinetic modeling and the in silico skin permeation model, which help forecast the drug absorption and permeation. And most recently, the in vitro laboratory-based experiments to define clear criteria for qualitative, quantitative, and structural similarity, as well as refine in vitro testing methods such as in vitro release testing and in vitro permeation testing, are proposed for demonstration of bioequivalence. The research finding of these in vitro approaches informs the development of product-specific guidelines that streamline the regulatory approval for generic topicals, and it ensures that they are closely match the reference product in terms of sensory attributes, composition, and drug product performance. And of course, this PSGs based on this research can provide patients with more affordable options while maintaining safety and efficacy standards. So indeed, the physicochemical and structural similarity or sameness are critical for bioequivalence performance of topical drug product. And as stated in the Code of Federal Regulation, any ANDA may include different inactive ingredient provided that the applicant identifies and characterizes the differences and provide information demonstrating that these differences does not affect safety efficacy of the proposed drug product. And the agency have released several product-specific guidance that recommend bioequivalence approaches of the products that show no significant difference in formulation to the reference standards. Most recently, the FDA released a draft guidance on the physicochemical and structural characterization of topical drug product that is stating what may be the criteria that used for Q3 characteristics. For example, the assessment of appearance, texture, investigation of phase status and structural organization of matter, and assessment of the polymorphic form of the active ingredient, and the rheological behavior characterization, and the characterization of water activity, drying rate, pH, and buffering capacity, and similarity in the oleaginous component and specific gravity, as well as characterization of any metamorphisms related to changes in the formulation as it is applied to the skin surface. So for example, here we can see the product-specific guidance for metronidazole cream that's released in October 2022. And the guidance is stating two options for demonstration of bioequivalence. The first option for bioequivalence study with clinical endpoint, and the second option is in vitro bioequivalence study and other in vitro characterization to demonstration sameness of Q1 and Q2 and Q3. And in particular about Q3, visual appearance and texture, phase status, structure organization of matter, rheological behavior, pH specific gravity is recommended, and more specifically, any other potentially relevant Q3 attributes in addition to in vitro release testing. Now research is needed on determination of what is these additional Q3 attributes that may be relevant to this specific drug product, and the reference standard is listed. Therefore, the in vitro physicochemical characterization and innovative research have played a critical role in shaping the FDA guidance for topical drug product. This influence has been seen in inclusion of physicochemical data, that informed the development of bioequivalence guidance for ensuring standardized and comprehensive assessment criteria. And also it had been seen in introduction and validation of innovative analytical technologies when backed by solid research. And also it has been seen in addressing the challenges of multiphasic formulation through extensive characterization and research, and also in real-world data from the physicochemical studies when it contributes to the integration of real-world evidence into the bioequivalence evaluation process, and it enhances the regulatory decision. And also it had been seen in continuous guidance improvement based on scientific advancement and stakeholder feedback, with data from physicochemical studies contributing to this continuous improvement. The last but not the least is that the agency always aligns with international guidelines through physicochemical approaches that contributed to harmonizing the regulatory standard globally. So these approaches not only support the FDA's commitment to ensuring safety and efficacy and quality of generic topical drug product, but also it fosters the international harmonization efforts and then benefiting the patients, healthcare providers, and pharmaceutical industry. So now let's go over a couple of FDA physicochemical research studies that supported the BSG development for topical drug products. The first case study of bioequivalence assessment of terbinafine topical ointment using in vitro studies. Terbinafine ointment 1% contains mono and diglycerides and propylene glycol as inactive ingredient. The optical images showed insoluble phases within the ointment matrix. This insoluble phase may be attributed to the insoluble portion of the mono and diglyceride, and of course, further investigation was then performed to explore the chemical identity of this dispersed phase of the formulation and to determine the drug distribution among these phases. So the research included microscopic examination, staining test using different dyes, and then chemical identification using Raman spectroscopy. And the technique that was used, it was morphological directed Raman spectroscopy. To determine the chemical identity of these insoluble structures, a library of Raman spectra for each individual component, as well as for the insoluble fat separated from the matrix, were done. The Raman spectra were compared, and the results showed that the spectra of the matrix and this insoluble structure are the same. In addition, propylene glycol showed the highest correlation in both the insoluble fats and the matrix. So this study confirmed that these insoluble structures are insoluble fats of mono and diglycerides rather than immiscible droplets of any other inactive ingredient. And the API terbinafine is mainly distributed in the continuous phase and to a lesser extent in the dispersed oily structure of the ointment. Therefore, this ointment may be regarded as a single-phase ointment for other bioequivalence assessment studies. And based on these results, a product-specific guidance of terbinafine topical ointment was released in October 22nd. The PSG does not recommend the IVBT here for phase status. However, it recommends characterization of visual appearance and texture of the ointment, as well as characterization of phase status and structure organization of matter using microscopic examination with representative high-resolution microscopic images at multiple magnification, among other in vitro characterization methods for demonstration of bioequivalence. The second case study included bioequivalence assessment of clascoterone topical cream using several in vitro studies. Clascoterone topical cream is a multiphasic formulation made of several hydrophilic and hydrophobic excipients and surface active agent. In vitro permeation testing may be then used to assess the dynamic distribution of the active ingredient among different phases of the formulation, and to study how this distribution may affect the continuous absorption and availability of clascoterone. It is worth noting that clascoterone molecules are not stable in the physiological solution and can be hydrolyzed to cortoxylone via cortoxylone 21 propionate at body temperature. Therefore, this study aimed at assessing the feasibility of conducting IVBT for clascoterone cream in assessment of its bioequivalence and to determine what is the appropriate analyte for comparing the rate and extent of clascoterone bioavailability after topical administration. For this, the research included developing an analytical method using liquid chromatography with tandem mass spectrometry for quantitation of clascoterone, cortoxylone, and cortoxylone 21-propionate in IVPT and mass balance samples. And the tool that had been used is liquid chromatography with tandem mass spectrometry. The chromatographic analysis of the permeation sample was done without any immediate sample processing, and the chromatogram of the 24 and 36-hour samples showed a significant decrease of clascoterone solid blue peaks and a significant increase of cortoxylone 21-propionate white peaks within 12 hours period. However, the internal standard peaks intensity was stable over this observed period. The permeation profile of clascoterone and cortoxylone was then obtained using the inline flow-through system with constant removal and replenishment of the receiver solution. This in vitro permeation profile of clascoterone and its metabolites were obtained over the course of 48 hours. So this test was performed using a dermatome skin samples from 4 donors, and the IVPT cumulative permeation and flux data demonstrated good reproducibility and similar trend of clascoterone permeation among all donors. And the highest flux of clascoterone permeation appeared over about 6 hours in all donors. The outcome of this study supported a revision of the product-specific guidance of clascoterone topical cream, and based on this study, IVPT testing was feasible and may be used to characterize the effect of globule site distribution of clascoterone topical cream and its topical absorption. And clascoterone and cortoxylone may be used as analyte for permeation samples. These analytes were observed as early as 2 hours in the receptor solution, indicating metabolism of clascoterone during IVPT testing. And timely sample processing as the test progressed could prevent post-permeation metabolism and loss of clascoterone in the permeation sample. The guidance revision is scheduled and it will be released this month. So the key takeaways from these slides is that pharmaceutical research supports the development of BSGs for assessing bioequivalence of generic topical product, and it provides evidence for safety, efficacy, and therapeutic interchangeability between generic and reference topicals. And comparative characterization of the physicochemical and structural properties of complex topical product is a part of the comprehensive assessment for pharmaceutical equivalence, bioequivalence, and product approval. And as the product complexity increases, so do the risk and the need for more comprehensive Q3 characteristics. And the research-driven evidence ensure approval of safe and effective generic topicals, and indeed it benefits the patient having affordable and reliable medication. Let's go to a challenge question. Which technique may be used for providing morphological information and identifying molecular components through chemical mapping of topical formulations? A. Scanning electron microscopy. B. Differential scanning calorimetry. C. Fourier transform infrared spectroscopy. And D. Morphologically directed Raman spectroscopy. So the answer is D. Morphologically directed Raman spectroscopy, as it combines Raman spectroscopy image and imaging to enable obtaining morphological information and identify the molecular component in multiphasic topical products through chemical mapping. So at the end of my presentation, I would like to acknowledge the team of topical drug product in both OBQ and OGD, all the names listed there so that I'm not forgetting anyone. And thank you for listening to my talk, and we can open more questions during the panel discussion. Thank you.